The first speaker is Bartosz ULASZEWSKI, and he's going to talk about spatial genetic structure of European beech and outlier SNP analysis of multiple European beech.
My presentation is quite short, so I think I will catch up. First of all, I would like to thank the organizers for inviting me to this wonderful conference. This presentation, the results were in collaboration with Joanna and Professor Burczyk, our boss. So I have to speak about him. I have to make one more remark that the numbers that you will see here and the abstract and in the final paper may be a little bit different, but the general results are the same. So don't stick to numbers, please. We are talking about European beech, probably the top three broadleaf tree species in Europe. It's a very widely represented. It has a wide range of representation, but its differentiation is among populations is generally low. Existing studies based on neutral markers show patterns of genetic diversity, for example, in allelic richness, heterozygosity, which are related to distance from glacial refugia. As we talk about climate change, forecasts predict, because the species still evolve, it still migrates, so it will migrate towards central Scandinavia, Baltic countries, Lithuania, and there are some forecasts that it may decline on areas where exhibit long-term drought events. So to be fair, we have this kind of situation in Poland, and I think in Ukraine, so we are just in the middle of a disaster. Okay, you have the map, so we have. Let's see. So in our study, we have used material from two common garden sites, one located in north, one located in central Poland. The common garden sites contained material from 85 populations. Some of them doubled, so the yellow dots are the doubled populations. The red one are from central common garden site, and the blue one are from the northern site. So you can see that most of them are from Germany and from Poland, but we have also few from France, from Spain, Italy, Romania, Czech Republic, and Slovakia, and from Denmark, of course. We have assessed those populations with neutral markers, SSR markers, just to double check them. And you all know that SSR markers are still used, so we want to have some like a comparison. But then our main goal was to assess those populations with ddRAD data. To be fair, we haven't used the reference genome that is deposited and available and published. We have assembled our own reference genome. But we are now reanalyzing it and checking the results with the reference genome 1.3 version, and we get the same results, to be fair. We have mapped the reads, called the SNPs with HEAP. It's not a very popular software, but it uses the same approach as GATK, so you can call it GATK. For the genome environment assessment, we have used the WorldClim data, and we have also analyzed the spatial structure, genetic diversity measures, and differentiation. I have to make one more remark that we have transformed the climatic data with PCA analysis just to reduce the redundancy. So PC1 and PC2 are factors responsible reflecting temperature, and PC3 and PC4 are factors reflecting the rainfall. Okay. To show you that this is quite okay, the PC1 factor reflects the continental Atlantic climate influence. So it's very good. So from our gigantic, quite big amount of SNPs, we have filtered them down to 12,000, almost 13,000 SNPs. We wanted to have like the SNP represented in over 95% of samples, minimal allele frequency over 5%. We wanted to assess allelic SNPs, and we have removed the indels. We have also indels there. To do the assessment, we have divided the SNPs to neutral markers and candidate SNPs under selection with BayeScan, LFMM, and Sambada. I will talk about this a little bit later. Yeah, thank you. In general, the neutral genetic diversity have been like similar between the SSR markers and SNP markers. The numbers are not the same, but you know, it's a different type of markers. But the SSR markers, as we can see, show like a very blurry picture. You can see some patterns here. We have also like a support for the correlations between the latitudinal, so like a north-south heterozygosity, but not support. We have not supported the previous studies with that. But with the SNPs, neutral SNPs, we have found like a very good picture. What is the migration routes for beech? So we have like a 3 clusters, main clusters. One from here, second from here, and third from here. Okay, so like okay, and here's a mistake in the presentation. Sorry for that. This graph shows the relation between FST and distance between the pairs. It's not much. It supports the previous studies. Now the interesting part, I think. The genome-environmental or PC analysis of SNPs. So from those 4,000 SNPs, we have like 150 SNPs that we are sure or almost sure that are associated with environment. Because we have used BayeScan, LFMM, and Sambada, and we have chosen only those SNPs which are detected by all 3 methods. You all know that there is a problem that we have a lot of candidates and a lot of outliers, but the true SNPs are very tricky to detect, so we wanted to be very conservative, and we have chose only 150 SNPs with that. Okay, so the genetic diversity, neutral versus adaptive, we can see that the populations in general show very, like here we have like a very dense, the range is not much, but here the range is very, very broad. Some populations exhibit very low allelic richness, heterozygosity, and very high FST. To be fair, we have also assessed the structure, the clusters, based on the PCA analysis, on the PC SNPs, and it is generally the same. So the SNPs are correlated with also with the neutral structure of the populations. Here we have we have put the heterozygosity the heterozygosity of the eastern populations is quite low, same as allelic richness. We have like a very high FSD in the eastern populations. So I think that we can say that the populations here are under threat because in the case of any dramatic events, I think that the population may be like the genetic diversity may be not enough to survive of these populations. But the good thing is that the Western populations are generally in good condition, so we are quite lucky. And I have to make a second remark that those populations here in Poland are a part of a nature reserve, so we cannot do very much to plant new trees and to get them. The FST here shows it's a very big, sorry, just too stressed. We have also assessed the SNPs, and we wanted to find if there are annotated with some sequences, with some coding sequences, and half of them are in the coding sequences or very near the coding sequences, so they have influence on the coding sequences. We are in the middle of analyzing them to analyze what they are responsible for at this moment. Okay, so quick summary. Our results provide evidence that the structure, additional evidence to previous studies about the structure of the species, about the genetic diversity of the species. We have identified 150 non-neutral SNPs, which are related to climate variables, and they demonstrate genomic background of the adaptive diversity. We think that the populations which exhibit very low genetic diversity must be monitored. So it's a very good tool to monitor them because they are under threat, under serious threat. I would like to acknowledge our collaborators from University of Poznań and Institute of Dendrology in Kurnik, Professor Barzda and Professor Roskowski, and our lab teammates Katarzyna and Eva. Thank you.
We have 5 minutes for questions, so Sylvie?
Hi, so Sylvie Muratorio in Rai, France. If I remember well, the study by Comps on recolonization based on isozyme, finally you have the same kinds of patterns for non-neutral SNPs as for neutral
adaptive
markers. Yeah,
we have the same patterns. Clusters are the same, but the genetic diversity is different in both of those assessments.
But the trends and the decreasing diversity in the eastern as compared to...
Yes, they are the same.
François Lefebvre from INRAE. Sorry, I didn't get how you resolve the confusion that there can be between the geographic distribution of climate and the geographic recolonization routes. How far can we distinguish markers of the recolonization routes and markers from climate, related to climate?
I think that the methods that we used are we try to be conserved, but we are not 100% sure that the problem is solved.
Santiago González from INRAE. You have a very clear pattern of isolation by distance. But I was wondering how then can you make groups for differentiated population to look for outliers? Because really you don't have clusters that are separate, but just a covariant. So I was wondering the underlying model for the testing for the outliers, if normalized isolation.
To be fair, some of those research are done not by me, so I try to answer I will try to consult your question with our lab team, and I'll give you an answer on the coffee break. Is it okay?
Okay, that's okay. Thank you.
Yes, my question was, maybe I missed it, but how similar was this geographic structure that you got with your neutral markers and with these SNP markers which are neutral and SNP markers which are related to genes? Was it similar or no, this geographic
structure? It was like the similarity was very high. So like I can say that share of cluster, for example, share of cluster, neutral cluster number 2, and share of cluster PCA cluster number 2 is like a 92%. So correlation is very high.
Hi, thanks for the talk. Of course. I'm Sean Hoban. I'm at the Morton Arboretum in the United States. You said that about half of your outlier SNPs were not in sort of the known gene parts of the genome.
Yeah.
This is a question that might come up for other talks throughout the couple of days. Do you think those are places that are not well annotated, or we don't know their function yet? Or do you think those are mistakes or false positives?
Very complicated question. I think that, okay, so with those SNPs, we have like annotated them like artificially. So we have extracted the sites and compared them with the data, with the available data sets. So I think that those sites are like, okay, maybe I can answer to the question in the coffee break. I'm sorry, I'm a little bit too stressed.
Okay. Okay, thank you. So